The pine wilt nematode, Bursaphelenchus xylophilus, was first discovered in 1971. This discovery occurred in the United States and has since been recognized as a major pathogen responsible for pine wilt disease, which has caused significant damage to pine forests in various parts of the world, particularly in East Asia. The route of infection by the pine wilt nematode, Bursaphelenchus xylophilus, involves a detailed and coordinated interaction with pine sawyer beetles, genus Monochamus, which act as vectors for the nematode. Adult beetles initially become carriers of the nematode by feeding on infected pine trees. These trees harbor the nematodes, particularly in branches and twigs. As the beetles emerge from their pupation inside pine trees, they carry the nematodes within their bodies. They then migrate to healthy pine trees for their maturation, feeding on the young twigs and bark. During this process, the nematodes are transferred from the beetles into the sapwood of these healthy trees. Once inside a new host tree, the nematodes leave the beetle's body and infiltrate the tree's resin canals. They spread through the vascular system, feeding on tree cells and disrupting the flow of water and nutrients. The visible symptoms in the tree, such as wilting, needle discoloration from green to yellow and then to brown, and rapid tree death, often occur within months of infection. As the infected tree dies, the nematodes reproduce and accumulate in the wood. New beetles that tunnel into the dead tree to lay eggs pick up these nematodes, thus perpetuating the cycle of infection. This complex interaction highlights the challenges in managing and controlling pine wilt disease. Pine wilt disease, caused by the pine wilt nematode Bursaphelenchus xylophilus, manifests through several severe symptoms that lead to rapid decline and death of affected pine trees. One of the first visible signs is needle discoloration. Healthy green needles begin to appear dull grayish-green or yellow and progressively turn brown as the disease advances. Concurrently, the trees exhibit signs of wilting, a direct consequence of the nematode disrupting the tree's vascular system, which is essential for water and nutrient transportation. As the infection progresses, the tree's overall health declines rapidly. Unlike some tree diseases that develop slowly, pine wilt disease leads to a swift deterioration, often resulting in the tree's death within weeks to months after the initial appearance of symptoms. Another notable symptom is a reduction in resin production, an indicator of the tree's compromised health due to the nematode's activity within the resin canals and vascular tissues. These symptoms, particularly when they appear together, typically signify an advanced infestation. Given the rapid progression and severity of the disease, early detection and prompt removal of affected trees are critical for preventing the spread of the nematode to nearby healthy pines. Managing and treating pine wilt disease, caused by the pine wilt nematode Bursaphelenchus xylophilus, requires a comprehensive approach involving various strategies to prevent, contain, and directly control the nematode. The primary method for immediate control is sanitation and removal, which entails the prompt removal and destruction of infected trees. This helps prevent the vector beetles from spreading nematodes to healthy trees, particularly if done before the beetles emerge in the spring. Chemical control using systemic insecticides can also be employed to manage beetle populations. This reduces the likelihood of nematode transmission but must be carefully managed to minimize environmental impacts. Alongside these methods, research into biological controls is ongoing. This includes exploring the use of nematophagous fungi and bacteria that target and inhibit nematodes, offering a potentially environmentally friendly alternative, although these options are still largely experimental. Developing and planting varieties of pine that are resistant to the nematode is a proactive long-term solution. Some species and hybrids have shown inherent resistance and are recommended for planting in areas prone to pine wilt. Supporting tree health through good forestry practices, such as proper watering, mulching, and protecting trees from mechanical injuries, also plays a critical role in enhancing resistance to diseases like pine wilt. Additionally, implementing quarantine measures and restrictions on the movement of pine materials from infected regions helps prevent the disease's spread to new areas. This includes regulating the transport of potentially contaminated wood, bark, and soil. These combined efforts are essential for effective management of pine wilt disease and require collaboration among forestry professionals, landowners, and regulatory agencies to ensure early detection and rapid response.